Good morning, ladies and gentlemen, and good evening to our honorable guests in the United States. Welcome to the 2021 Bilateral Symposium on Brain Research hosted by Ministry of Science and Technology from Taiwan and National Institutes of Health from the United States. In the symposium, we will have reputable researchers to share the outstanding results in brain science field with the three main topics, including imaging platform and brain atlas, neuromodulation of neurodynamics and behaviors, and clinical applications, prevention, diagnosis, and treatments of neurological diseases. In the beginning, we are very delighted to invite Dr. Hongchen Chen, Director of Department of Life Sciences, Ministry of Science and Technology, to give his opening remarks. Now, let's welcome Dr. Chen.
Ladies and gentlemen, good evening for the U.S. Entities, and good morning for the Taiwanese entities. So on the behalf of the Ministry of Science and Technology, I sincerely welcome all your participation to today's symposium on brain research. So we are very grateful to all invited speakers, moderators, guests, and colleagues, in particular Dr. Zhen Qing, who helped to organize today's symposium in a very short time. So US and Taiwan have had a long partnership on many aspects, including economics, culture, science, and technology. Innovative brain research technology are expected to have huge impact on not only medicine, but also industries. Many countries, including US and Taiwan, have launched programs to promote brain research. Today's symposium is going to provide a forum for distinguished scientists to share their insight and experience on three topics. First, image platform and the brain atlas. Second, neuromodulation of neurodynamics and behaviors. And third, clinical application. So I hope through our in-depth discussion, we can facilitate our understanding and promote the cooperation between US and Taiwan. So now let me briefly introduce the brain research program supported by the Ministry of Science and Technology. So the annual total budget for brain research is more than $20 million. We support brain research in Taiwan through 4 different channels, including a brain research center led by academician Professor An Shijiang at the National Tsinghua University. A brain technology strategic program, an R1 grant in the division of life science, in particular in the division of clinical neuroscience, biochemistry, and pharmacology, and finally, national core facilities. Okay. So the overall goal of Taiwan brain research is to solve the brain by developing advanced technology, mainly used for 3 aspects: brain atlas, neuromodulation, and clinical application. Among national core facilities in Taiwan, 5 of them are commonly used by our brain research teams, which include synchrotron radiation, biomedical, optical, and radiography, animal facilities, chip design, and the high-speed computing and storage platform. In 2017, the Ministry of Science and Technology initiated a 2-year brain pilot study. 2 years later, in 2019, the Ministry of Science and Technology officially launched a 4-year brain technology strategic program. After fierce competition, 12 cross-disciplinary research teams were selected for funding. Actually, the Taiwanese scholars who will present later in today's symposium are all supported by this program. So here I would like to announce that there will be another 4-year brain strategic research plan sponsored by the Ministry of Science and Technology from 2023 to 2026. The next phase of brain strategic program will be more emphasized on technology integration and the clinical application. So the Brain Research Center, led by academician Professor Anshi Jiang, is co-supported by the Ministry of Education and the Ministry of Science and Technology with annual budget around $4 million. This center used Drosophila as the model organism to develop innovative brain technology and to understand the mechanism of memory. So the Professor Anshi Jiang is a world-renowned scientist. So he's going to introduce more about Brain Research Center later. So to encourage the Taiwanese neuroscientists to collaborate with investigators at NIH, we provide extra $1 million per year to add on their brain project. So the amount of budget sounds quite small, but I have to say, this is an extra money. It's only for the beginning of our cooperation. So of course, if our cooperation is quickly expanded and show the strength, I believe the new strategy or mechanism for our bilateral cooperation will be soon created. So finally, I wish today's symposium a great success and all participants a most rewarding and enjoyable time. Thank you for your attention. Thank you.
Thank you. Thank you, Dr. Chen, for the opening. Coming up, let's welcome Dr. Julie Huayu from Institute for Translational Research in Biomedicine, Kaohsiung Chang'an Memorial Hospital, to share current execution and achievements of Taiwan Brain Technology Development and International Rising Program. Let's wait for her online a little bit.
Hello, can you all hear me? Hello, can you hear me? Okay. First of all, good evening to the delegates and fellow neuroscientists in Bethesda, and good morning to those in Taipei. I'm Julie Chen from Kaohsiung Chang'an Memorial Hospital. It really gives me a great pleasure to have this opportunity to introduce to our fellow scientists in the NIH, the Taiwan Brain Technology Development and International Raising program. In my capacity as a co-convener in charge of coordinating this program. Now let me share with you my screen. The Ministry of Science and Technology has a long-standing history in promoting brain research in Taiwan and to the globe. In addition to the regular just like Dr. Chen mentioned in his address, in addition to the regular ROI research grants, in recent years, MOST has launched top-down commissioned programs on brain and mind sciences. And the current program, the Taiwan BTDIR program, was launched in 2019. Goal of this program is to advance brain research in Taiwan by promoting formation of cross-disciplinary and innovative collaborative teams that strive for translational application of scientific achievement to our society. Like I said, the MOST has a long-lasting history in supporting brain research in Taiwan. And in addition to the regular R01 grants, in recent years, MOST has launched a top-down commission program to promote brain research. And the current Taiwan Brain Technology and Development and International Raising Program, BTDIR, was launched in 2019. May I have the next one, please? Goal of this program is to advance brain research, brain science research in Taiwan by promoting formation of cross-disciplinary and innovative collaborative teams that strive for translational application of scientific achievement to society, and to increase our visibility in the international community by increasing our global competitiveness. And the major missions of this program, just illustrated and outlined it by Dr. Chen in his address, is to conduct cutting-edge research in brain science from the molecular and cellular levels to neural circuits, and to integrate this multi-level information to the understanding of human behaviors in health and disease. In addition to that, we would like to play an important role of an international and national research hub in brain research, and to stimulate the interaction between scientific institutes, academia, and industry. So the framework for this program, may I have the next one, please, is actually 3 parts. The first part is to understand normal brain architecture and development and functioning, from synapses to circuits to behavior. And in addition to that, we like to address health challenges that arise from abnormal brain functioning through prevention, better diagnosis, and treatment. An ultimate goal of this program is to apply research outcomes to improve individual brain health and social and cultural well-being across lifespans. So based on this framework and the structure and foundation, may I have the next one? We implement several strategies to its realization. The first, of course, is innovative research technology. We like to develop innovative research technology to perform cross-field. It's not just biomedical. We also include a natural science, social science, etc., for their collaborations in attempt to commercialize the research outcomes and to stimulate national and international collaboration. And all this are actually inspired by 2 major notions. May I have the next one? The first one is from Sidney Brenner, who said that progress of science depends on new techniques, new discoveries, and new ideas, probably in that order. So we like to focus on the new technology as the foundation to the understanding of the brain function. And another one is breakthroughs often come from bringing in tools and concepts from other fields and apply those to your problem. This is by Bruce Alberts. So based on those notions, this is why we set these 3 major missions of this program. And currently, we have 12 projects that are granted under the current program. For the purpose of the presentation, we grouped the projects into 3 categories, although many of the projects actually cover more than 1 categories. May I have the next one, please? The first one is imaging platforms for spatial temporal mapping of brain functions. These include advanced optical imaging platform for mapping brain functions, mapping the human brain connectome by bridging micro to meso to macro organizations, decoding pain sensitivity in migraine, with multimodal brainstem-based neural signatures and recovexial rate nonlinear optical microscope to unravel brain connectome and signaling. And one of them will be presented in today's symposium. And the second group, may I have the next one, please, is neuromodulation strategy. And these include innovative precision neuromodulation system for mechanistic investigation of cortical basal ganglia circuit dynamics in treating Parkinson's disease, mapping, deciphering, and modulation of memory engrams. This is a multidisciplinary approach, and this topic, again, will be presented in today's symposium. And in addition to those, we have focused ultrasound modulation as a new tool to explore brain functions and apply this in treatment of neurological diseases. And the third group is, can I have the next one, please? It's actually the translational application for all this basic and bench work and their scientific information for prevention, diagnosis, and treatment of neurological diseases. And these include brain aging, which will focus on muscle-to-brain axis, to particular to modulate physiocognitive functions, and the intelligence adaptive deep brain stimulation system for precise treatment of Parkinson's disease, neurological basis of soreness. We would like to launch a new concept for this sensation called sensation and its therapeutic potential for neurological disorders. Of course, treatment of Alzheimer's disease by overcoming energy crisis is a new angle to the treatment of this degeneration disease, and decoding and promoting resilient brain from multivariant profiling to intervention. And the last one will again be presented in this symposium. In addition to this, the presentation that you will hear in a minute, the others, because of time, we didn't invite them to have the chance to talk on their research work, but all of them have made a very prominent research progress, and would like to particularly highlight three. And I'd like to take this opportunity to apologize to the rest of this project and to colleagues in Taiwan that we didn't have time to touch upon every project. So the first one is, may I have the next one, please? Is the focused ultrasound modulation for epilepsy suppression based on EEG observation. This team led by Professor Haoli Liu from Department of Electrical Engineering in National Taiwan University have found effective focused ultrasound parameter for epileptic spike suppression. And they have histologically confirmed that neuron can be activated through this focused ultrasound neuromodulation, and the brain is intact after ultrasound exposure. And they can modulate at a molecular level of some protein expression and protein phosphorylation. And this technology has been proved by IRB for clinical trial currently. May I have the next one? The next example is the project led by Professor Wang in the Taipei Veterans General Hospital. He's a physician as well as a neuroscientist who developed an intelligence system of migraine detection and neurostimulation for assessing treatment. The wearable brain-computer interface system can combine with an AI predictive migraine attack system, and they can translate that to direct current stimulation technology to help migraine patient in treatment and in diagnosis. May I have the next one, please? And the third one is the project led by Professor Mingdao Ke from Institute of Electronics in National Yangming Jiaotong University. His team has developed an adapted closed-loop deep brain stimulation system with an embedded system on chip design that is in good compliance with medical electrical equipment standard and has been verified in animal model of Parkinson's disease. So those are the ones that I'd like to briefly introduce to you. But if you're interested in any of the project, I think we can have mechanism for further discussion. May I have the next one, please? Another important mission of Taiwan BTDR program is to play the role of a national and international research hub in brain research. For this, we plan to implement this by enhancing national and global networking through the establishment of collaborative network by joining Global Brain Research Initiative. May I have the next one? The IBI was officially launched on November 3rd, 2018 at the Society of Neuroscience meeting in San Diego. And the organization is composed by a number of major initiatives around the globe, including, of course, the Brain Initiative in the US, the Human Brain Project in the European Union, Australian, Canadian Initiative, China, Japan, as well as Korea Brain Initiative. And the stated goal of this organization are to serve, may I have the next one, the scientific community and provide them with a forum for information sharing to build resources for global brain research collaboration in a coordinated global brain research effort. So I'm very happy that we open our dialogue with our colleagues and delegates and fellows, neuroscientists in the NIH, paving way for global collaboration and partnership in this IBI organization. So I look forward for very active interaction, and thank you very much for your attention.
Thank you, Dr. Julie, for the kind introduction. Next,
I'm so sorry for the technical problem and hiccups during my presentation, and thank you for your patience.
It's okay. Thank you very much. Next, let's welcome Dr. Anshi Zhang from Brain Research Center, National Tsinghua University, to take us through the current execution and achievements of the Brain Research Center. Welcome, Dr. Zhang.
Good morning and good evening. It's a pleasure to introduce the Brain Research Center to you. The center is composed around 50 different laboratories across more than 10 universities and research institutes, focus on one single question, that is how the brain acquire, learn, and remember an experience. And hopefully we can learn from that to develop the smarter AI. Sorry, over there. Let me start it to introduce our past experience. We started to ask this question 20 years ago from an invention that making the entire fruit-fly brain transparent. Once the tissue becomes transparent, all the internal structure become very clear. And using this technology, we start to map olfactory circuits in the higher brain centers and study how where and exactly which individual neurons that synthesize new proteins to form long-term memory. And then study how the information travels through different pathways and shunting from a pathway to another pathway based on the differences in the information content. And more recently, we developed a series of new technologies, which I will introduce some of those to you later. Basically, we are trying to visualize every single molecule in the entire fruit fly brains at extremely high resolution near to the EM resolutions. So, as I say, that the centers composed of many different interdisciplinary expertise. I'll skip this. And the first direction is to map the entire Drosophila brain at the synapse level. Okay, the entire Drosophila brain has around 130,000 neurons, so it's really more doable. Okay, and we systematically to map one at a time, and now we have collected about 90,000 individual neurons, and compared to the EM work performed by the Genelia and Google and Wellcome Trust, they have recently released around 20,000 individual neurons, Drosophila connectons. Of course, EM provides the synapse resolution, and light microscopy does not provide that. So in the past few years, we focused on this and trying to reach to the EM resolution using light microscopy, and that allowed us to quickly complete the entire brain connections. And in addition, we start to use the map to study function. Can you click on this? Oh, sorry. Can you click on the? Yeah, and this one. Yeah. So these are the calcium functional imagings, because one way we kind of catalog every structure is to keep the brain within the head capsule. So this in situ templates allow us to map not only the structural connectivity, but also to compare that with function activity. So a group of people then focus on developing a series of new technology to improve each of those important aspects of functions, and to understand the brain activity. In short, that now we can stimulate any cells in the entire fruit fly brain and look at the response at the entire brain, every single neuron in the entire brain. And another group of people working on computational connectomics, and once we get this image, it developed, we have automatic segmentation methods allow us to segment individual neurons and predict the priority, and then compose to predict the information's flow in the entire brain, and trying to understand the codings and to simulate how the brain functions. And we focus on understand memory and tracing one neuron at a time. And this is olfactory projection neuron. It delivers odor information to this processing center here. And once it reaches there, it kind of associate with punishment or reward, and the information start to stored through new protein synthesis at this downstream output neuron. And we then trace along the neurons, trying to identify every single neurons that synthesize new proteins that change brain's function. And more recently, we think this is still too slow, and we are focusing, trying to develop the so-called next generation connectomics. And as I explained to you, currently early this year, Genelia and Google announced Hemi-Brain's project's results, and with EM's resolution. So we started by using another technology called expansion microscopy, developed by Edward Boyden at MIT. And so you can see that this is a fruit fly brain, and expanded 10 times in diameter, it looks like this. I will show you later, we can zoom in and looking at every individual neurons. I will also show you another independent approach using synchrotron's X-ray microscopy. So in short, that we are able to now expand not just 10x, but 40x. This tiny dot here is the real fly brain, okay? Once it expanded 10 times, it then looks like this. Expanded 40 times, looks like this. The little fruit fly brain at around 1 to 3 millimeters it's 1 to 3 centimeter. And once you expanded this, we can now, can you click here? Yes, okay. Embed it in a gel and then image the whole brain automatically and cut off the imaged part and then continue to finish all the image. And the results then automatically stitched into the entire brain. And once you focus, then you can see the individual neuron fibers. And in theory, 40X will be around 8 nanometer resolution. So I'll show you this expansion microscopy at 10X expansion. This is entire brain's dopaminergic neuron. It's like Google, you look at all the street views and then zoom back to see the entire brains. So what can we do with this? So one of the study is to look at individuals' Bhutan's plasticity. This is EM resolution. Okay, this is our 10X expansion. So this is a single Bhutan. It looks very close in terms of resolution now, but it's much, much faster than EM, okay? So you can imagine that this is a single Bhutan in the entire fruit fly brain. So you can calculate and measure every single Bhutan here. Each color is a single Bhutan. And then we just being able to show that this morphology of the Bhutan is activity dependent by manipulating its activity individually in the entire brain without affect other neurons. So independently, we develop a series of new technology using X-ray. And so if this is human brain, fly brain is like this. And we brought it up, and using synchrotron, we can now directly visualize individual neurons in the head and reconstruct the entire body, in fact, in 3D, and develop the new technology, allow us to see gene expressions using X-ray. I believe this is probably the first time that someone see this under the X-ray at resolution close to the regular confocal microscopy. Oh, this is not working. Okay, this is last slide. So because of these new technologies, it is fast and extremely high resolution. We think now we are ready to move from fruit fly, which we are most interested to understand learning and memory. But this technology can be readily applied to larger animals, including human brains. So we're looking for collaborations with hospitals and also these computer centers. Okay. And this is just started. We don't have data yet, but we think the new technology allow us to map the large brains at the synapse level finally. I will stop here, and thank you very much.
Thank you, Dr. Zhang, for the fascinating presentation. After the introduction of Taiwan's current achievements, we are really honored to have Dr. Nina Shore, Deputy Director and Acting Scientific Director, National Institute of Neurological Disorders and Stroke, National Institute of Health, to give us the introduction to NIH Neuroscience Program. Warm applause to welcome Dr. Shore.
Thank you all very much, and good morning to our colleagues in Taipei, and good evening to my colleagues at NIH. It really is an honor to be able to introduce you to neuroscience on the NIH campus. As you've already heard, the NIH campus is in Bethesda, Maryland, and all of the buildings that house neuroscience are designed in such a way as to have many of the 27 institutes that are part of the National Institutes of Health represented. So neuroscience really is not a single department or a single entity on the NIH campus. It really is a transdisciplinary enterprise. Okay, so what you are seeing now is the Porter Neuroscience Building. And the Porter Neuroscience Building houses basic science. It is primarily laboratories, and they are contributed to by many, many of the institutes of the National Institutes of Health. This is a picture of the clinical center at NIH. And this entire hospital complex is dedicated to clinical research. And so all of the institutes that do research that involve either outpatients or inpatients or studies that need to be done on specimens from patients use this building as the site for their research. And now our newest building on campus is on the next slide. And this is currently a temporary building that will house the Center for Alzheimer's and Related Dementias, which is really a joint effort of the National Institute on Aging and the National Institute of Neurologic Disorders and Stroke. But again, all of the institutes at the National Institutes of Health are contributing to the research that goes on in this building. On the next slide, you will see what it is that characterizes neuroscience research at NIH, and that is that it tends to be high-risk, high-reward research. NIH funds the investigators on its campus for their entire research program, not for individual projects. And for this reason, investigators don't write a separate grant application for each project that they do. They are funded for somewhere between 3 and 4 years' time, and they are free to take the kind of risks that allow you to do high risk and high reward research. The mechanism for translation from the bench to the bedside, from basic research to translational research to clinical research, is quite facile, and training programs right on our campus fuel the next generation workforce. So the next slide gives you an idea of the kinds of research that we do on the NIH campus. They go all the way from the brain to the spinal cord to the peripheral nerves to the muscle. And from a mechanistic standpoint, they go from basic to translational all the way through clinical. And because all of the institutes of NIH contribute, they involve the nervous system control of other organ systems, neuroendocrinology, neurology of aging, integrative neuroscience, clinical neuroscience, things like neural development and the changes that occur in the nervous system from childhood through adolescence through adulthood. The next slide gives you an idea of the kinds of neuroscience that occurs in each of our institutes. And each of these letter abbreviations, each of these acronyms symbolizes a different institute at NIH. And so the National Institute of Neurologic Disorders and Stroke does basic and clinical neuroscience. The National Institute of Mental Health does psychiatry-related neuroscience. The National Institute on Aging does neuroscience related to aging, geroneuroscience. National Institute of Child Development does developmental neuroscience, and so forth. And so neuroscience really pervades the NIH campus, and it currently is the most highly funded science on the NIH campus. It just recently overtook cancer in its allocation of funding. And much of what goes on in the National Cancer Institute is very relevant for neuroscience as well, brain tumors and peripheral nervous system tumors. The next slide really gives you a sense of the productivity of the institutes relative to neuroscience. The National Institute of Neurological Disorders and Stroke alone comes out with approximately 400 publications annually. And one of the things that I think most of us enjoy the most about neuroscience on the NIH campus is that we have students and trainees at just about every level you can imagine, from summer students, most of whom are college level students, through graduate students, post-baccalaureate students who spend a year or two with us, postdoctoral fellows, and clinical fellows who do their clinical training in the context of clinical research. And you can see that despite the pandemic, our numbers for all of these have remained relatively stable. There was in 2020, our summer student program was curtailed for the safety of the students. But by 2021, we went back up to pretty close to what we had originally done using telecommunications. And we run many successful training programs on the campus. And I dare say it's one of the most enjoyable parts of the neuroscience community. So I'm delighted that we are joining you this evening and this morning for exploring the potential for our collaboration and for mutual involvement in fueling the next generation workforce in neuroscience. So if I could have the final slide, I just want to thank all of you for your colleagueship and your partnership, and I'm very much looking forward to hearing more about neuroscience in Taiwan. Thank you.
Thank you, Dr. Shore, for the abundant information. We are very glad to have you and your colleagues here. And kindly ask you, Dr. Shore, can you please keep your camera on with us, please, because following up is our photo call session. So today we are honored to have so many reputable speakers and guests in brain science to join this symposium. To commemorate this important moment, we will take a group photo all together. And that's first welcome the online speakers. Please turn on your camera and wave your hand to our audience when I call upon your name. So let's welcome Dr. Nina Shore, Deputy Director and Acting Scientific Director, NINDS, NIH. Dr. Mark Hallett, Chief Medical Neurology Branch and Human Motor Control Section, NINDS, NIH. Dr. Yeka Appointee, Associate Director, Diversity and Inclusion, National Institute on Drug Abuse, NIH. Dr. Luigi Ferrucci, Scientific Director, NIA, NIH. Dr. Susan M. Resnick, Chief Laboratory of Behavioral Neuroscience, NIA, NIH. Dr. Julie Huayu, Institute for Translational Research in Biomedicine, Kaohsiung Changgen Memorial Hospital. Dr. Shulan Xie, Department of Psychology, National Chenggen University. Dr. Joshua Goh, Graduate Institute of Brain and Mind Sciences, National Taiwan University. Dr. Yang Fan, Director, Intramural IT and Bioinformatics Program, NINDS, NIH. Dr. Shirley Yang, Director, Science and Technology Division, Taipei Economic and Cultural Representative Office in the U.S. These are our online speakers. Furthermore, we have many distinguished on-site speakers. Let's welcome Dr. Hongchen Chen, Director General, Department of Life Sciences, MOST. Dr. Yihan Sun, Institute of Molecular Biology, Academia Sinica. Dr. Anshi Jiang, Brand Research Center, National Tsinghua University. Dr. Zhenzhang Lian, College of Life Sciences, National Yangming Jiaotong University. Dr. Xiwei Zhu, Molecular Imaging Center, National Taiwan University. Dr. Mingkai Pan, Department and Graduate Institute of Pharmacology, National Taiwan University College of Medicine. Now I'd like to invite our online and on-site speakers, please raise your right thumb for us and give us a thumb up for the future development of brand technology industry. Thumbs up, please. And our online on-site speaker, can you please put on your mask? We all have two formats. The first one off, and the second one with your mask. Thank you. Okay, again, thumb up, please. Under the strong support of MOST and MIH, we believe through this symposium we could exchange the current status and achievements of both sides. Thank you. We thank greatly to all attendee speakers. So, in the following session, we will discuss 3 topics, including imaging platform and brand atlas, neuromodulation of neural dynamics and behaviors, and clinical applications. For the first session, we will have discussion on imaging platform and brand atlas. Let's welcome Dr. Xiwei Zhu from Molecular Imaging Center, National Taiwan University, to present spatial temporal mapping of brand functions. Welcome, Dr. Zhu.
Hello. Good evening to our friends in the States and... feel much better. So good evening to the friends in the States and good morning to the friends here in Taiwan. So I'm Shihui Chu from Physics Department in National Taiwan University. It's really my honor to give a talk here, and I think that as a physicist, I'm really honored to be founded by this Taiwan Brand Project. This truly displays the interdisciplinary nature of this Taiwan Brand Project. So I work together with Dr. Mingkai Pan in Medical School of National Taiwan University to like today, I'm going to talk about spatial temporal mapping of brain functions. Following the talk of previous speakers, I believe that every one of us understand this is important to understand the brain. I think this is one of the most important scientific challenge in this century. And how to understand brain, I think, in my opinion, this is actually similar to try to understand a computer. When we want to understand computer, it is not enough to understand hardware only, but then what inside a computer. Similarly, when we want to understand the function of brain, as Professor Zhang just showed up, this is a state-of-the-art electron microscopy connection map of a Drosophila brain. So this is hardware mapping of a brain. But to truly understand the brain, we certainly need to understand also the software inside the brain. And software can be defined as program and operation information used by a brain. Toward this goal, to understand the software of a brain, we certainly need to map the function inside the brain. So this is also mentioned by Professor Zhang that both structure and functional connectome are very important. So the outline of this Taiwan Brain Project, as mentioned by Professor Julia Hua-Yu before, that this has 3 major directions, including imaging platform, neuromodulation, and the clinical applications. For imaging platform and neuromodulation, I think many of us are focusing on functional and structural connectomes. In particular, for neuromodulation, is more focused on functional connectomes, and we hope to use this connectomics knowledge to clinical applications. And I'm in charge of one of the projects in the imaging platform called Advanced Optical Imaging Platform. So I'm going to introduce the structure of this optical imaging platform. So here we focus on technology development, in particular optical imaging, optical microscopy. So we work together with researchers from several different institutes, different disciplinary, and also different countries toward developing the technology of optical imaging, including contrast, resolution, speed, and depth. In addition to that, we work together with information scientists to establish smart analysis platform to understand the brain function. And with this innovative tools, we also work together with biomedical researchers to apply these tools to understand brain functions as well as brain diseases. Oops, excuse me. Okay, right, so due to time limitation, I think I'm going to show some of the key results in this project. One is starting from structured connectome. We work together with Dr. Bi Zhang Chen in Academia Sinica to develop macro light sheet microscopy. The idea is to use a camera macro lens to acquire very large area, very large field of view and high resolution image. And as you can see here, this is the image of a whole mouse brain, and we can really see the whole brain as well as individual cell inside a brain. And this is a whole mouse embryo, so that we move from whole brain connectome to a whole body connectome with this innovative large area imaging tools. And as Professor John mentioned, by expansion of the brain, we can get even higher spatial resolution with this structured connectome. And in terms of functional connectome, as I mentioned, I think this is one of the key steps toward really understand the brain. We work, the key technology here is high-speed volumetric two-photon imaging. So we start a few years ago from single beam, very high-speed volumetric imaging. So with single beam, the volumetric imaging reaches about 1 to 10 hertz, 1 to 10 hertz, and this is the image of a whole Drosophila brain, and we can observe in real time the calcium response inside. To move forward to a mammal imaging, we also combine with endoscope and this volumetric imaging, so that it allows us to capture arbitrary region, volumetric region inside a living mouse brain. So this is an example of individual cell functioning inside a whole SCN region, which is located at the bottom of a mouse brain. And more recently, we expanded this technology from single beam toward 32 beam simultaneously scanned together. So this allows us to really boost up the imaging speed from a few volume per second up to 100, more than 100 volume per second. So now we can map very high speed volumetric imaging in a living brain. And we were contacted by, I put this paper on archive, and we were contacted by Dr. Yuanyuan Liu from NIH, who really want to collaborate with us on this high-speed volumetric imaging of brain. And the next step we are looking forward in the near future is to further expand the number of parallel scanning beams to boost up the imaging speed further. And in terms of application, we have now very good tools for large tissue imaging, very good tool for functional imaging. And so we work together with medical doctors to explore the possibility of using these tools to study disease and as well as to understand the mechanism of a disease. So here we work on essential tremor. We'll work on essential tremor with Dr. Mingkai Pan together. This tremor is most common movement disorder, but the mechanism is largely unknown. So based on the knowledge from Dr. Pan, he told us that the drug response of essential tremor, such as this drug propranolol, is less than 50%. So the treatment of this disease is not really effective. And the mechanism of behind this essential tremor can be several different origins. Two of them are climbing fiber overgrowth and also hormone-induced tremor. This hormone can be found from barbecue, which we have a lot of them in Moon Festival here. And interestingly, both these mechanisms induce action tremor. Action tremor means that when the patient or the mouse tremor occurs. So Dr. Pan create animal models based on these two different mechanisms and observe both action tremors in them. So here we are going to apply a few technologies to understand the mechanism as well as the treatment effectiveness of essential tremor. So the first technologies we're using is high-speed two-photon imaging so that we can monitor neuron firing. These are Purkinje cell neuron firing in vivo when the mouse is freely moving under the stage. And working together with image information scientists, that we have smart data analysis that can categorize these functional neurons into different groups And by analyzing these responses, we found that for the fiber overgrowth mouse, the Purkinje cell firing has persistent synchronization behavior, while for Hamelin-induced tremor, the Purkinje cell synchronization is action dependent. Like here, when it is moving, you see more synchronized behavior. So please remember that they are similar in terms of expression. So more interestingly, we also work together with Dr. Zhongxuan Luo in simulating the neuron response. So he established a simple model including excitatory and inhibitory neurons and very beautifully reproduce a similar behavior that when the structure of the neurons are based on climbing fiber overgrowth, he observed very strong synchrony in the response, while it is in a different mechanism, he observed moderate synchrony. So this agrees quite well with experimental observation. So to link this animal model behavior, animal model observation to human being, Dr. Pan carried out a cerebellar EEG to observe the response inside the brain. So this is still model mouse behavior. So you can see here that from these two mechanisms, this climbing fiber overgrowth and Hamelin-induced tremor, the oscillation pattern are very different in these two mechanisms. More interestingly, these two mechanisms has different response to this drug for essential tremor. So for this climbing fiber overgrowth, the drug response is less sensitive. For Hamelin-induced tremor, the proponal response is much better. So these are currently all model animal response. So the real interesting result is here that when we combine the result with human, because cerebellar EEG you can carry out in human being, so we observe very similar behavior in human being. That in the EEG result, it can be separated into two different kinds. One is persistent oscillation, one is action-dependent oscillation. So it is in line with the animal behavior observation. And these two different groups also has different response to this drug. So we believe this model animal study can be derived all the way to a human disease response. So in summary, we are very happy to be part of this Taiwan Brain project as an interdisciplinary and international project. And the three emphases include the imaging platform, neuromodulation, and clinical applications toward the establishment of functional and structural connectomes in brain. So in our own project, we are working on optical imaging platform, including structured connectome based on high resolution, very large field of view imaging, as well as functional imaging with very high speed and whole brain or whole brain region imaging inside a living mouse brain. And by working together with information scientists, we can work on smart analysis and circuit simulation to further understand the mechanism behind a disease. So we work together with medical doctor toward clinical applications, and here we give an example of essential treatment. So this is a review paper that I wrote very recently, and I hope this can be a nice reference for not only researchers, but also students in NIH. Thank you so much.
Thank you, Dr. Chu, for your talk, along with those amazing images. Secondly, we will have Dr. Mark Hallett from Medical Neurology Branch and Human Motor Control Section, NINDS, NIH, to elaborate more on non-invasive brain stimulation methods, focusing on possibilities for Parkinson's disease.
Right. Hello. I hope everyone can hear me. I am pleased to have the opportunity now to speak to colleagues in Taiwan in the morning and to my friends here in Bethesda in the evening. And I'm going to talk about non-invasive brain stimulation, focusing a bit on Parkinson's disease. I have a little bit of a conflict of interest, as does NIH, because we hold the patent for the H-coil, which is a type of coil for non-invasive brain stimulation. Brainsway has licensed this coil, and it is now FDA approved for certain treatments. but I'm not going to be talking about the H-coil in this particular lecture. So in terms of the mechanisms of brain stimulation, there are basically two in order to get a good clinical effect. One of them is an online effect. That is to alter brain function with a lesion, anatomical or functional, that would interrupt a brain circuit or modulate the oscillations within a brain circuit. And this is what one does with lesion surgery or deep brain stimulation. And this, of course, is very effective in a number of different circumstances, including different types of tremor and Parkinson's disease, and has really revolutionized treatment for various neurological disorders. But another is persistent effects. One can modify the brain using brain stimulation by inducing a plastic change of some kind. And this is what we do with non-invasive brain stimulation. Here we use the non-invasive brain stimulation and hope to have a long-lasting effect for a therapeutic benefit. Now, there are many devices for non-invasive brain stimulation. Probably the oldest such device for clinical purposes was or is electroconvulsive therapy, ECT, which is now being modified to some extent to use magnets rather than electricity, called magnetic seizure therapy, transcranial electrical stimulation, transcranial magnetic stimulation, static magnets, transcranial direct current stimulation, transcranial alternating current stimulation, transcranial random noise current stimulation, temporally interfering electric fields, focused ultrasound, and transcranial electromagnetic treatment. Many different devices, and many of these are being used in different laboratories at NIH. I'm going to focus on transcranial magnetic stimulation, which is one of the tools that has been most popularly used in recent years. So how do you make a plastic change? As I pointed out, for a non-invasive stimulation, one wants to have an intervention and then have a long-lasting benefit for the patient. Well, first, there can be direct effect of transcranial magnetic stimulation itself. TMS, used in different patterns and different forms of repetitive stimulation, can lead to a direct plastic change. Second, TMS can prime the brain so that a second intervention can be more effective. So, for example, one can prime the brain and then give a second intervention, such as physical therapy or training, and that could augment the benefit of the training. Now, there are a number of different mechanisms for priming, and this is a very interesting area of basic plasticity research, both in animals and humans, trying to understand how brain stimulation can prime the brain for more optimal change going forward. And there's a mechanism called gating. There's a mechanism called homeostatic plasticity. There's another one where, interestingly, when one tries to relearn something that is learned before, the old engram actually gets disrupted and then becomes reconsolidated. And this is something that priming can do as well. So there are a number of different mechanisms, and how one can use these optimally is not yet completely clear. Now, I want to note that TMS after training can disrupt consolidation. So this is not a good way of using non-invasive brain stimulation, but priming does appear to be particularly useful. So there are many different patterns of repetitive transcranial magnetic stimulation, as I mentioned earlier. One can just simply use repetitive transcranial magnetic stimulation, fast and slow. Fast seems to facilitate brain increase excitability, slow decrease excitability. Theta burst transcranial magnetic stimulation, continuous and intermittent. Continuous tends to suppress and intermittent tends to excite. Quadrupulse, TMS, and paired associative stimulation, which is a form of heterosynaptic plasticity. But these latter two haven't been used as much for therapy as the first two. One other thing about making a plastic change in the brain, treatments need to be repeated multiple times to get a substantially long-lasting effect. And one could consider this similar to practicing a new skill. Repetitions are needed to drive an enduring plastic change. Now, therapy with repetitive transcranial magnetic stimulation of various kinds is used in a wide variety of indications. Perhaps most successfully at the present time in psychiatry. The ones that are in red here are the ones that are approved by the FDA. Treatment of depression, obsessive compulsive disorder, addiction, and migraine headache. But it is being used for a variety of other things as well in different experimental settings. And I'm going to focus now on the treatment of Parkinson's disease. And I'm going to particularly deal with treatment of Parkinson's disease gait. Parkinson's disease gait is a particular problem in patients. It is very difficult to manage, even with other therapies. So dopamine replacement and DBS, of course, are the common uses, the way that people treat Parkinson's disease. They can be helpful, but for Parkinson's disease gait, it doesn't work as well as for other elements of Parkinson's disease. And note also that the efficacy of these drugs and this technique is useful only when used. There's no long-term effect. Physical therapy can be potentially beneficial. Balance training and practice walking, for example. And other popular activities can help, such as dancing or boxing. But I'm going to be talking about non-invasive brain stimulation. Now, there are a number of studies that just use direct non-invasive brain stimulation to alter plasticity. And these have had some success for sure, but the benefit hasn't been as good as one would like. And so I'm going to turn to the issue of potentially using brain stimulation for priming and then doing training. Now, one of the first good studies in this regard actually came out of Taiwan. This was a paper that was published in 2013, and I am very pleased to note that a number of the co-authors of this paper are former fellows of mine at the NIH. Dr. Lau and Dr. Cheng were former fellows. And in this picture of a time that I visited Taipei in 2019, here we have Dr. Lau and Dr. Cheng. And here's Dr. Pan, who was featured for his work with essential tremor in the last talk. So going back to this paper, the authors did do what I mentioned, to combine TMS and treadmill training to see if they could improve walking. And they compared treadmill training by itself to treatment with 5 hertz TMS first and then the treadmill training. So priming was in fact particularly valuable for this. So let's just look, for example, at control subjects that just had treadmill training and experimental subjects that got the repetitive TMS first. Let's just look at comfortable walking speed, although other parameters showed similar changes. Just by training, one goes from 108 centimeters per second to 114 centimeters per second. So treadmill training can help. But if you do priming, you go from the same, about 108 centimeters a second, now to 123 centimeters a second. So a much larger benefit from using priming. Now, in this situation, the assessment was done just after the training, so we don't know how long it lasted. I was involved with another study that pursued this matter further, and this reports a paper that was published in the Annals of Neurology last year. We had 45 participants with Parkinson's disease randomized to 3 groups getting TMS prior to treadmill training, rapid RTMS at 25 hertz, slow RTMS at 1 hertz, and sham. And the reason for using 25 and 1 was to try to understand a little bit better the mechanism of priming, since this facilitates the cortex and this tends to inhibit it somewhat. So we did 12 sessions of RTMS and treadmill training performed over 3 weeks, 600 TMS stimuli at 80% of the motor resting threshold were delivered to the leg area of the motor cortex of each hemisphere. And these are the results. This is looking at the increase in fastest walking speed. The dotted line here is what happened with sham TMS, so it's basically treadmill training alone. And you can see that both of the priming techniques caused a much larger increase in the fastest walking speed. This is immediately after training. This is 1 month later, and this is 3 months later. So even 3 months after completing the training, there was a persistent effect in increase of fastest walking speed. For the timed up and go test, which is another measure of improvement, this benefit was seen only with the 25 hertz, not with the 1 hertz. The 1 hertz was similar to the sham stimulation. So you can see that priming plus training may well be more successful than RTMS alone or training alone. There is no ill effect of homeostatic plasticity. This is one of the types of priming that we were considering, and we were thinking that homeostatic plasticity might have an ill effect, but it did not. And gating or reconsolidation, those other methods, which were utilized by the 25 hertz, may be the most effective intervention. So just to conclude then, non-invasive brain stimulation can be a useful therapeutic tool. There are a variety of mechanisms of plasticity that might be operative. In some circumstances, it might be useful to use 2 interacting mechanisms, such as priming. And of course, there is still much more to learn in this particular situation. So thank you very much for your attention. I am pleased to have had the opportunity to bring this information to you.
Thank you, Dr. Hallett, for your sharing, and we hope to see you again in Taiwan soon. In the next session, we will discuss neuromodulation of neurodynamics and behaviors. Let's welcome Dr. Chen Zhenlian from College of Life Sciences, National Yangming Jiao Tong University, to give speech on hippocampal cell types and circuits that mediate avoidance behaviors. Welcome, Dr. Lian.
Good morning and good evening, everyone. Welcome to this session, Neuromodulation of Neurodynamics and Behavior. My name is Zhenzhang Lian from Institute of Neuroscience of National Yangming Jiao Tong University. Under the framework of most Taiwan Brain Project, there are 3 groups working on neuromodulations. As shown here, Dr. Yu Wei Wu's group is developing an innovative precision neuromodulation system for mechanistic investigation of cortical basal ganglia circuit modulate dynamics and treating Parkinson's disease. Dr. Liu Haoli's group is developing focused ultrasound neuromodulation as a new tool to explore the brain mechanism. My group is using an interdisciplinary approach towards mapping, deciphering, and modulation of the memory engram. Generally, my group and the Aponta group at NIH have common research interests. We are interested in discovering principles of how neural circuits perform computations underlying complex behaviors such as emotion and cognitions. Moreover, we are interested in developing technologies to explore or modulate the neural dynamics in defined cell types. Our research focuses on 3 key brain regions in the limbic systems, including the hippocampus, amygdala, and hypothalamus. These key brain regions mediate our survival behaviors, including avoidance behavior and feeding behaviors. We are currently determining the roles of neuronal circuits that mediate these behaviors. More recently, we are interested in determining how these neuronal circuits are involved in nociception and their potential as a target for pain treatment. It is generally believed that our emotions and cognitions are no more than the behavior of a vast assembly of neurons, so-called neuronal ensemble. For instance, lesion studies have demonstrated that the hippocampus is important for memory encoding. However, an emergent and fundamental question is, how do specific cell types and neuronal circuits give rise to complex behaviors? Today, my talk is going to focus on how hippocampal cell types and circuits that mediate avoidance behavior. Over the last decade, scientists have successfully uncovered the input-output organization of the hippocampus at the structural levels. However, how information is processed after entering the hippocampus remains largely unclear. In the past 10 years, my group have identified several subtypes of GABAergic interneurons, including local and long-range GABAergic cells in the dentate gyrus. We have identified and determined their circuit mechanism that can control the output of the principal neurons, that is the granule cell, and regulate their excitability. So speaking of this input-output dynamics of the hippocampus, I need to talk about the importance of GABAergic interneurons. So given the importance of GABA interneuron in regulating the hippocampal dynamics, I'm going to focus on this specific type of cells called MOSI cells. MOSI cells are glutamate-ergic neurons in the dentate gyrus. MOSI cells receive excitatory input from the granule cells and mediate the feedback and feed-forward excitation onto the granule cell and the local circuit GABAergic interneuron, respectively. Interestingly, MOSI cells project their axon to the contralateral counterpart of the dentate gyrus, and they regulate the excitation inhibition balance and control the hippocampal dynamics. So recently, using the GCAM6 based calcium imaging, we discovered that these MOSI cells are preferentially active during open-arm exploration. So this is a test for anxiety-like behavior. The mouse is exploring in this open arm. This is called elevated primates, which is a kind of approach avoidance test. As you can see, when mouse spans in the open arm, the calcium signal is increased dramatically. When the mouse turns to the closed arm, you can see the calcium signal is gradually decreased. We also combine juxtacellular recording and optogenetics to determine the circuit of MOSI cells. As you can see here, elevating MOSI cells can recruit GABAergic interneurons in the dentate gyrus and suppress the granule cell, the DG output and finally the CA1 output. We next determine that elevating MOSI cells can suppress the anxiety-like behaviors in the next slide. So using chemogenetic approach, we can selectively activate the MOSI cells in these freezing animals. As you can see here, on the right side is elevating MOSI cell activity using chemogenetic activation. On the left-hand side is the control study. As you can see, the animals spend more time in the open arm compared to the control group. So here is the summary of the trajectory of animals. You can see elevating MOSI cell activity decrease avoidance behavior. Taken together, we have demonstrated that MOSI cells are preferentially active in anxiogenic environment. Activation of MOSI cell results in GABAergic interneuron activation, thereby reducing EG and the CA1 output. We also demonstrated that elevating MOSI cell activity decrease avoidance behavior. Next, I want to use a few slides to talk about our recent common interest in nociception and chronic pain. As you can see here, this is the input-output organization of the amygdala. The amygdala, or the central amygdala, is a nociceptive amygdala. This central amygdala receives nociceptive input from the brainstem. In juxtacellular recording, we recently identified two types of central amygdala neurons. These two types of neurons mediate the nociceptive response. As you can see here, we identify pain on or pain off nociceptive input. This is also called pain activating and pain suppressing central amygdala neurons. Interestingly, we found that these two types of central amygdala neurons undergo opposite change in their semantic excitability and semantic plasticities. So using optogenetic techniques or chemogenetic approach, or the drug, the clinically approved drug, pregabalin or Lyrica, we can reverse this rewired central amygdala neuron and ameliorate the pain and the negative emotions. So here is a slide showing that the pregabalin or Lyrica can suppress the central amygdala neuron activity and improve the negative emotion and hypergeusia in chronic pain mouse model. As you can see that at the baseline, you can see these fresh neurons in the central amygdala neuron. 40 minutes after pregabalin application, you can see there is a great reduce in the number of these fresh neurons. So finally, I would like to thank my team members for their contribution, and also I want to thank the Taiwanese founding agents most, and also my collaborators, including Dr. Aponta at NIH. Thank you very much for your attention.
Thank you, Dr. Lin, for the detailed explanation. Coming up, let's welcome Dr. Yeka Appointee from Neural Circuits and Behavior Unit, Associate Director, Diversity and Inclusion, NIDA NIH, to talk about hypothalamic cell types and circuits that drive survival behaviors.
Good morning, good evening, everyone. I am very delighted to be here today virtually and tell you about the research in my lab. So my lab is generally interested in the mechanisms by which brain circuits regulate natural or survival behaviors, such as food intake and nociception. So when I think about the major epidemics of our generation, besides this ongoing global pandemic, two issues come to my mind, which are obesity and the opioid overdose crisis. And just to give you an update on the statistics, these are the numbers for obesity and the mortality rates due to opioid overdose in the US. So to me, these numbers are truly devastating, as I can see that these problems are not only affecting people in the US, they are global epidemics and a major cause of death. Therefore, my lab is determined to understand how neuronal circuits drive the rewarding and addictive nature of food, but also identify circuits that modulate nociception and their potential as a target for pain treatment. We're starting in the hypothalamus because it's known for decades to be a brain region that regulates survival behaviors. As you know, the hypothalamus is one of the deepest structures in the brain in both humans and mice. We use mice as our animal models. Pioneering studies show that the hypothalamus regulates feeding, drinking, mating, sleeping, circadian rhythm, aggression, you name it. Moreover, it is massively connected to brain regions involved in reward and goal-oriented behaviors, such as the ventral tegmental area, the amygdala, and the nucleus accumbens. And furthermore, it is so rich in terms of genetically identified cell types, making it ideal to study the neuronal basis of behavior. Therefore, in order to make sense of this lateral hypothalamic chaos, my lab members and I are committed to understand and determine how the genetically identified cell types and neuronal circuits drive behaviors that are essential for survival. So today, I'm going to tell you about two genetically distinct cell types that, even though they are located in the lateral hypothalamus, they have very different functions when it comes to survival behaviors. These are the lectin receptor expressing neurons and the parvalbumin positive neurons. We are studying the role of the lateral hypothalamus as a brain region gating reward, appetitive, and consummatory behaviors. In particular, we aim to determine whether lateral hypothalamic LEPR neurons, which happens to be GABAergic, contribute differently to appetitive and consummatory behaviors. In order to address this question, we use transgenic mice and viral strategies for cell type specific neuronal manipulations, together with optogenetics and chemogenetics. But as you can see on these schematics, we also needed to develop and use functional imaging techniques to record the activity of multiple neurons deep in the brain in vivo and during behaviors. In these schematics, you can see the expression of GCAM6 in the lateral hypothalamus and a green lens chronically implanted and fixed to the skull with dental cement to record those neurons using a single photon miniscope in freely moving mice. Now, we also designed a chronically implantable guide cannula in collaboration with Lodoric lenses, which allows for the insertion and removal of green lenses across session. Now, by using this approach, together with a 2-photon system, we were able to visualize GCAM6 positive neurons in the lateral hypothalamus with subcellular resolution, as you can see on this image. In this example, we use a 2-photon scope to image changes in fluorescence from GCAM6 positive neurons in the lateral hypothalamus during anesthetized and awake states, as you can see in this video. Now, every trace is color-coded and depict the number of neurons recorded in the region of interest. So neural activity was measured by the calcium transient changes in fluorescence, also known as delta F over F, and this is represented on these traces. Now, we found that LH-LEPR neurons respond to appetitive stimuli in vivo during a Pavlovian conditioning paradigm in which an auditory cue predicted sucrose reward. So what you see in this graph is that the activity of LH-LEPR neurons increases during the tone, indicating the delivery of sucrose compared to the tone with no reward. And furthermore, acute optogenetic activation or inhibition of LH-LEPR neurons did not affect food intake, as you can see on this graph depicting the amount of food consumed before and during photostimulation or photoinhibition. So together, our results demonstrate that LH-LEPR neurons drive appetitive but no consummatory behaviors in mice. All right, at the beginning of my presentation, I quickly mentioned that we're trying to make sense of this lateral hypothalamic chaos. So now I'm going to switch gears and tell you about the other cell types, which are the lateral hypothalamic parvalbumin positive neurons. So throughout the central nervous system, parvalbumin positive neurons are typically GABAergic interneurons with fast spiking phenotype. However, we previously showed that this small cluster of neurons, around 150 cells per hemisphere in the mouse, are located in the very lateral side of the lateral hypothalamus. They are fast spiking, they are glutamatergic, and they send long-range projections. But are they necessary to drive behaviors essential for survival? So we show, using optogenetics, that these neurons modulate nociception. What you see on this graph is the latency to port withdrawal during photoactivation or photoinhibition of LHPV neurons on mice that have been placed on a hot plate. What we observed was activation of these neurons attenuates thermal nociception or decreases pain, while inhibition promotes nociception. We also show that these neurons respond to a noxious stimuli in vivo. So for this, we use functional imaging and genetically encoded calcium indicators and tested the mice for thermal nociception, in this case on a hot plate. And what we observed was a cluster of neurons that responded to the thermal stimulus, as you can see on these traces and also on these heat maps, which is consistent with the role in modulating nociceptive processing. Now, I would like to emphasize that this is the first time that the activity of LHPV neurons has been recorded in vivo and during behavior. Now, what about translation and clinical applications? So by using chemogenetics, we found that these neurons could modulate nociception on a prolonged time scales, as you can see on this graph of thermal nociception during activation or inhibition across 3 days. Now, I truly think that some of the pain models that I have shown you in mice are crucial for a mechanistic understanding of pain modulation at the cellular and molecular levels. However, they could be challenged when it comes to translation and clinical applications as they measure pain-evoked behaviors. And as we know, we very seldom see humans walking on a hot plate or being poked by a bone fry filament. As a matter of fact, one of the major problems for individuals dealing with chronic pain is that at some point they're not able to perform the daily activities. Therefore, we decided to test models of pain-suppressed behaviors. In this case, pain-suppressed nesting behavior in mice. We all know that nesting is an innate natural behavior in mice, so previous studies have shown that when mice are in pain, they are not able to put together their nest, so we induce visceral nociception by IP injection of acetic acid and measure the mice's ability to collect these little pads and put them together as a net. So what you can see on these schematics is that all groups were able to put together their nest during controlled conditions. However, that ability was disrupted after inducing visceral nociception, but then it was beautifully recovered by chemogenetically activating LHPV neurons. Now, in collaboration with the lab of Dr. Chenchang Lian, there in Taiwan, who happens to be one of my former graduate advisors, we would like to identify brain regions and neuronal circuits that provide excitatory inputs onto these LHPV neurons, and the neuronal circuits that provide and the neuronal circuits mediating the cognitive aspects of pain. So with that, I would like to thank my lab members for the passion and dedicated work, especially during these very challenging times, to my collaborators in the extramural world, and thank you all for your kind attention.
Thank you, Dr. Appointee, for the inspiring talk. So far, we have covered the two areas of imaging platform and brain atlas, neuromodulation of neurodynamics and behaviors. And we believe we all got abundant information after the remarkable speeches in the last two sessions. And some of our audience may have some questions, but don't worry, because we have arranged a 30-minute joint discussion later on in the end of this symposium. Please feel free to bring the question out until then. We are looking forward to discussion and exchange in the panel discussion session. If you have your question at your hand, you may take a note and write it down first. The next session will be clinical applications, prevention, diagnosis, and treatment of neurological diseases. And for this area, of course, we also invited a lot of renowned scholars and researchers to share with us. Now, let's welcome Dr. Shulan Xie from Department of Psychology, National Cheng Kung University, to give us the overview of clinical applications. Welcome, Dr. Xie.
Hi, hello everyone. I'm pleased to have this privilege to participate in the third session of the symposium, Clinical Applications. As you can tell from this figure, half of the ongoing projects supported by the Taiwan Brain Technology Development and International Raising Program are devoted to clinical applications. Next. These projects, next slide, cover the topics of normal aging, pathological aging such as disability and dementia, mental disorders such as depression, neurodegenerative disease such as Parkinson's disease, and chronic pain. Next, the common feature across these clinical projects is to integrate multimodal and multi-levels of functioning data, including genetic, epigenetic, molecular, brain imaging, behavioral, physical, socioeconomic, and cultural levels. Next, the goal is to identify biomarkers for diagnosis, develop early intervention program for prevention, and innovate effective treatment methods for neurological diseases. Next slide. We are aware that the recent initiated precision medicine, next, considers individual variability in genes, environment, and lifestyle for each person. Therefore, we believe native Taiwanese data and research are urgently needed. Next, for example, some research has found that even a person showing the presence of pathological tau and beta amyloid in the brain, the person does not necessarily develop severe brain atrophy and cognitive dysfunction due to some complex multivariate factors, as mentioned before. This is conceptualized as brain resilience and cognitive resilience, and this is also the topic that our project is doing. So next, therefore, we would like to propose establishing a Taiwan comprehensive and longitudinal database. Although there exist numerous large-scale databases exploring aging trajectories, next, such as Baltimore Longitudinal Study of Aging, which will be introduced later by Professor Susan Resnick, the sample of Taiwanese is still a minority in the scientific literature, as they are dominated by the West countries. Next, the advantage of having our own Taiwanese database is to help identify culture-sensitive mechanisms that are operational across lifespan and develop more accurate treatment and prevention strategies for a particular disease. So next slide, I would like to invite Dr. Josh Gu from National Taiwan University to demonstrate how cultural differences may modulate aging brain and cognitive functions. Thank you.
Thank you, Dr. Xie, for the overview introduction. After we have a better understanding about clinical application, we will welcome Dr. Joshua Go from Graduate Institute of Brain and Mind Sciences, National Taiwan University. And he will talk about cultural differences in aging brain cognitive functions, as Professor Xie has introduced. And I believe there are much about to share, because it is the aim of this symposium to bring together those who have contributed over the period of years to brain-related field. Let's welcome Dr. Go.
Good evening to my colleagues in the US and morning to all my colleagues here in Taiwan. I'm very pleased to be able to share this research topic that's been very close to my heart. It's what I've been doing for the past 20 years. So this work, actually, I'm very privileged to have been able to do some of this work also in the NIH, in the BLSA, and I'll share more about this later. So can I have the next slide, please? Yeah, so one of the key questions that we've always been asking in aging is that whether we can assume that aging trajectories is universal. Do we all age in the same way? Because if we do, if biological aging wins, then there's really nothing much to do in terms of research, right? We just sit and wait for things to happen in that sense. But if there are actually factors in our life, in our lifestyle, that kind of like influence the way that we age, then this is a clue that maybe this is something that we can leverage on and then use to help us age better or avoid certain pitfalls. So we're very interested in using culture as a way to investigate whether aging is universal. If you grow up or if you if you have different experiences over your lifespan. So as a matter of convenience, a lot of studies we've examined differences between Westerners and East Asians. And this is really in terms of defining culture, this is really more for scientific convenience. We are not by no means saying that that's all there is to culture, that there's only Western or East Asian culture. And we also acknowledge that even in Western or East Asian culture, there's quite a lot of heterogeneity and dynamic cultures going on, right? But as a scientific practice, we often want to be able to use something that's easy to control and measure. It just turns out that one of the easiest cultural dimensions to investigate applies to the difference between Westerners and East Asians. So just so to kind of define things a little bit, when we talk about Western culture, we note that Western culture is typically characterized by a value for independence, being an individual, and then this is typically also associated with the more analytic cognitive style, which is what we call object focus. So in other words, for example, if you were to look at a cat, a Western or analytic style would involve focusing on the eyes or the color of the cat. It has fur, the features that define the cat. But in East Asian culture, the value is more for interdependence. We're more collectivistic. And so this is typically associated with a holistic point of view or context-focused cognition. So here you might look at a cat and you might note that the cat does not exist on its own. It exists in its context. It's maybe in a room during this time of the day, as opposed to other times of the day. So this contrast, this analytic, holistic contrast, has been characterized as a different predictive strategy that East Asians and Westerners have developed in our minds and in our brains because of the environment, the social environment at least, that we grow up in. So next slide, please. So that definition has been going around a lot in the humanities and all that. So as a scientist, we want some objective proof that these cultural differences in cognition do exist and that we can measure them, right? So one of the earlier studies that we did was to simply give Westerners and East Asians, in this case, the Westerners were from the US and the East Asians were Singaporean Chinese. And we just let them look at pictures like this. We did some special manipulations, but in general, it's about how Westerners and East Asians move their eyes when they look at a picture, as you see in front of you. So what I show here in the red lines and the red circles is a sample eye movement by East Asians. And as you can see, what an East Asian would do when looking at a picture like that is to focus on the eagle and then maybe move around a lot in the background. Whereas a Westerner in blue would focus a lot on the eagle and move to the background a lot less. And also note that the fixation's duration, which is coded by the size of the circle, for East Asians, you tend to just focus very quickly on one point and then move to the next. And whereas the Westerners will tend to focus on one point and then stay there a little bit longer, dwell there a little bit longer. So this is very characteristic of a holistic versus an analytic viewing strategy in terms of what your eyes think, what your eyes predict would be important when looking at a scene. And note that this is passive viewing. We didn't ask subjects to do anything special, just look at it. So this is sort of like the default strategy that East Asian and Westerners engage whenever we're looking at things, right? So just imagine, this is something quite fundamental that just in terms of our eye movements and the way our eyes kind of search out information in visual scenes is already so different. So you can imagine then how that effect might be amplified through the downstream processes in the brain, higher level things like memory or attention or executive function and inhibition, things like that, right? Right from the start already perceptually, things are different in terms of eye movement. Next slide, please. So we had that in terms of the eye movement, and then my colleagues and I, in my graduate doctoral work, we were very interested in, does this translate to actual brain indices, right? Does this mean anything in the brain? And the short answer is that it does. So what we did was to show subjects the stimuli on the left. So you get quartets of pictures of objects and backgrounds, and sometimes we selectively repeated the object or selectively repeated the background or both of them. And using this paradigm, we're able to isolate brain areas functionally that were sensitive to the object or the background or the binding of both. And in the middle panels, you can see on the top rows are the young adults that we scanned in the MRI, and the older adults in the bottom row that we scanned. And in the left column or the middle slices are the US subjects, and the right of them are the East Asian subjects from Singapore again. And the key thing is in the younger adults, both US and East Asians show nice segregation of object processing regions in the occipital area, background processing regions in the parahippocampal place area, and then binding of these two streams of information in the medial temporal areas. And the interesting thing is if you go to the older adults and then you focus on the US group, the older US people, they still show this segregation. It's a little bit reduced, and that's not surprising, with aging processes. But the key point is that it's there. You see the nice object background and binding. But and this is the surprising thing that really shocked us, is that in the older East Asians, the older Singaporeans, we only saw a background kind of response. We didn't see any object response at all. And this was really shocking to us. And in fact, when we saw that, we were really worried that our scanner had broken down because the objects are really salient, as you can see in the stimuli. So, we thought, let's give the older adults, older East Asians a chance. And so we called some of them back, and it's the rightmost column that I'm talking about now. We called some of them back, and we now conducted a separate experiment where we let them see the same stimuli, but now we explicitly instruct them to attend to the object and ignore the background. And in this case, in the top brain slice on the right column was the 10 of the subjects that we got their original data from passive viewing, which shows the background area only. But when you tell these people to attend to the object area pops back up. So they can see the object. They just don't process it as objects by default. At least that was our interpretation. So this squares very nicely again with the fact that there's a holistic strategy that's driving the way that older East Asians view the scene, so much so that their brain doesn't treat the objects as a separate thing from the context. And this obviously would have implications for binding in terms of how the hippocampal functions in older East Asians and how we might measure it. Next slide, please. So this is very complicated, but I'll walk you through slowly. So the previous studies we conducted focused on ventral processing, perceptual processing, and medial temporal associative binding processes. And then now recently in our lab, and also in collaboration with Dr. Resnick's lab, previously my postdoc, we are now focusing on the other part of the brain, the frontal cortex and the striatum, and how this interacts with the medial temporal regions, and whether this might show individual differences and cultural differences as well. So to probe the frontal processes, we had to engage a different paradigm. What we did was to use the lottery choice task, as you can see in the top left. It's a very simple task. You just show people how many points you can win, and then your probability of winning and the probability of losing, in which case you have to pay or sacrifice the number of points, right? And so in this case, it's kind of like gambling. So people will have to choose if they accept those stakes. So in the example here, it's 54 points with a 79% chance of winning it, you should accept it, or most people should accept that stake. And then we show them feedback and outcomes. So this is the lottery choice task. And we vary the expected value, the probability and the magnitudes of the stimuli. So when we scan young and older adults, when we put them through the behavioral task in the scanner, you can see in the middle, just to the right of the lottery choice task, is the acceptance rates for younger adults in orange and older adults in blue. And you can see that when the probability of winning is very high, about 90 or 70% chance of winning, everyone accepts it. But the key difference for older and younger adults is that when the probability is 50 or less than 50, younger adults don't accept that risk at all. It's a losing gamble, right? But we get at least 20% or so of older adults who would accept a losing stake of 90% chance of losing. So we view this as really interesting because it kind of suggests they think of 90% chance of losing as a 10% chance of winning still something, right? That would be the only kind of way we could justify accepting such gambles. And to the right of that acceptance graph, you can see the DT, the violin plots. That's just basically coding the same behavioral data, but in terms of decision thresholds. The key point to demonstrate that is that younger adults tend to have a very narrow range of individual differences in their decision thresholds. But you can see that the older adults range is very stretched out and tends towards risk-taking, which is that the decision threshold is lower. They accept gambles at lower stakes. So if we go to the brain data on the lower left panel, there's a very drastic, this is done in Taiwan, there was a very drastic difference in the neural circuitry engaged. You can see that the younger adults, when doing the lottery choice, they just use the nucleus accumbens. The striatum seems to be, driving this, or at least very sensitive to the differences in value. But the older adults, they don't use the striatum, or at least we didn't detect much of it on average. But instead, there was a very different network. Older adults showed value sensitivity in the medial temporal cortex, in the ventral medial prefrontal cortex, and also in the dorsolateral prefrontal areas. And you can see also that it's not just all a positive association, which is would be the orange voxels, like higher activity to higher value. But in older adults, we also get higher activity to lower value in the dorsolateral prefrontal cortex that's coded in blue. So it's a very different shift. It's a very dramatic shift of the neural systems engaged when older adults do the lottery choice task. So that's comparing young and older adults in Taiwan. And then I go back to the data that I was very privileged to be able to work with in the BLSA in NIH already with Susan, where on the right side, we also did this task, a modified one, a more simplified one, but we could get 173 people. And in these older adults, these were all older adults, we noted the very big range of decision types in older adults. At least 1/3 of older adults are risk-taking, and then 1/3 are risk-averse, and then you get some in the middle. It's really an interesting question then for us in the future as we continue this work. Is this indicative of cognitive decline in terms of individual differences of susceptibility to clinical pathology? And we see that signature also in the medial temporal regions, the striatal regions, and the medial frontal regions as is depicted in the lower right panel. So the next slide, please. So that was comparing frontal, medial, and medial temporal responses in young and older adults, and noting that there are tremendous older individual differences. And the question is back to culture. Can we define this as a function of culture in terms of the differences in value-based processing in subjects? To date, we have some collaborative data that's published with Angela Gutches at Brandeis University as well. And so far, we have the younger adult data. We're still working on the older adult data, but the younger adult data here, they did the same lottery choice. And you can see at the bottom left that the Taiwan younger adults have kind of like, you would say, a more closer to rectilinear kind of function. In other words, Taiwanese tend to say, as long as it's greater than 50%, I'm going to just accept it. And if it's lower than 50%, I will reject it. But the US younger adults, they have a more discriminative, a more smooth S shape. In other words, a 64% chance of winning is different to a Western younger adult than a 70% chance than an 80% chance. There's more discrimination there. And again, that seems to be very characteristic of a holistic kind of a process where everything is just a win-lose context versus a discriminative probabilistic encoding in which every feature of probability is distinct. And this is it's very important to note that the cultural difference for this behavior is captured in the ventral medial prefrontal cortex. And for those of you in the Alzheimer's literature in humans, we note that that is one of the areas of amyloid and tauopathy that does occur. And we see that in our older adults as well. So definitely this is an area of focus. So next slide, please. So those were all data on brain functions, and that I hope I've convinced you that there are functional differences due to cultural backgrounds. Of course, we haven't done a study where you can transplant people in different cultures and do a longitudinal thing, but that's the evidence we have so far. That's in terms of function. And finally, we asked whether does that actually lead to structural differences? And the answer again is yes. So on the left, we have young Westerners and young East Asians. Westerners have thicker frontal gray matter, and East Asians have thicker temporal gray matter. This has been replicated in several other studies by other groups as well. So that's in younger adults. And in older adults on the right, we see these cultural differences in cortical thickness replicated in older adults, which means even with biological aging, where you get cortical thinning and loss, atrophy, and things like that, the cultural differences still persist in terms of the brain structure. And critically, these were only observed in older adults who had higher cognitive ability. Older adults who expressed lower cognitive performances, they didn't have any more cultural differences. So we really think that in that case, biology won in terms of decline and sort of wiped out the cultural differences that might have been there in terms of their brain structure. Next slide, please.
Excuse me, Dr. Goh, in the interest of time, I'm afraid that could you please wrap up your presentation in a few sentences?
Yes, absolutely. That's the last slide.
Oh, okay, thank you.
Yeah, so this is just my kind of like a summary model of what we think is going on, that the brain has to incorporate social, cultural contexts as well as the physical information. But I'd like to highlight the information that's in the yellow box, kind of like all this evidence that does point to us that cultural factors are very important for understanding how our brain ages and our individual and mental health. So the implications are obvious for us to better specify and get more accurate aging norms. And obviously, this will be very important for diagnosis and treatment if we want to move forward. Can I have the last slide just to acknowledge the collaborators here? So yeah, there's a lot of people in all this work, both current and past. And I thank you for your attention.
Thank you, Dr. Goh. Thank you. That's a very interesting angle to look at our cognitive functions. And I can see that Dr. Goh is enthusiastic on his research work. And actually, this is the kind of passion and enthusiasm can be seen on all the scholars and researchers today who have attended this symposium. Okay, and then next, we'd like to welcome Dr. Luigi Ferrucci from National Institute of Health, and he's from NIA. He's going to share parallel physical function and a cognitive decline with Asian. And then we will move on to the next speaker, which is our last speaker, Dr. Susan M. Ransnick. Okay, so now please join me in welcome Dr. Ferrucci.
Thank you very much for this invitation. It's truly a pleasure and an honor to be talking to you. And I really see that the level of the presentation is very high. And I'm going to share with you some of the work that we have done in the Baltimore Longitudinal Study of Aging that is relevant to this discussion. I just want to tell you that the Baltimore Longitudinal Study of Aging is a high collaborative program. It's probably the longest longitudinal study of aging in the world, and there are many, many opportunities for scientists around the world to contribute and to participate in the use of this data. What I'm going to talk to you today is Can you have the next slide? What I'm going to talk to you today is a different perspective in the pathways to dementia. So when we talk about dementia, we always think about the typical progression, a long progression over many, many years, where we have accumulation of A beta and then tau, and then change in the brain structure, and then probably one of the first neuropsychological parameter that is declining is memory, and then we have a strong decline of clinical function. However, more recently, the next slide, next, just to make it simple. We have seen Arnold, go back. That you're going too fast. Can you go back to the previous slide? Okay, stop here. So more recently, we have started to realize that cognition is not the only element that is declining during this process. And so there is declining mobility with large heterogeneity across individuals. And so there is declining mobility that sometimes occurs very, very early before we see any other clinical sign of cognitive function, during the cognitive function, and late in the process. And so it is really important that we understand, especially in the course of the aging process, whether this association with the decline of mobility represent a subgroup of this population evolved through dementia through a specific pathway. The next slide is a meta-analysis that really show that there is a strong cross-sectional association between slow gait and impaired cognition. The next slide. And then this has been confirmed strongly in longitudinal data. So the association from the observation epidemiological perspective is there. Next slide. So in order to study better this association between cognitive decline and physical function decline, in collaboration with Teresa Tian, we used data from the Baltimore Longitudinal Study of Aging, and we classify individuals among those that were user aged, where we didn't see over the period of time, or on average 7 years, any decline in cognition and mobility. Those that show only memory decline, those that show only gait decline, and those that show parallel decline in memory and gait, that I will call from now on dual decliners. And why this is important? Because that really underlines a group of the population where the cognition and the physical function decline at the same time. And when we did the next slide, when we did a meta-analysis, we found that truly the dual decliner had a risk of developing dementia. There was synergistic between the two conditions, and that was very, very high compared to the other groups. And that also convinced us that there is something very peculiar and very specific about this group, and we wanted to study this group more in depth. The next slide. So one of the things that we wanted to see is what really happened at the level of the brain. And we found, if you look at the right part of this slide, we found that brain volume change for the dual decliner versus others really affected both sensory motor integration and locomotion, with the superior frontal gyrus, superior parietal lobe, precuneus, thalamus, and cerebellum that tended to be strongly associated with being dual decliner versus others. So there is some specificity in the localization of the damage. The next slide. And then we found that the relationship is in fact very complex, because not only the walking speed and the mobility predict the development of cognitive decline, but it's also true vice versa, In fact, the individual that had deposition of beta amyloid compared to those that did not have an over the threshold deposition tended to have accelerated decline of mobility. So there is really a complexity of association that was present here. And the problem is, what is the pathophysiology of this condition? Should we identify this as a group that potentially has also a different type of intervention that is necessary to prevent dementia? The next slide. The next slide is, in order to study this association, we started to do large metabolomic studies. And we looked at target metabolomics, looking at about 500 metabolites in plasma. And we found that the dual decline was mostly associated with a decline instead of an increase with aging of a specific molecule. These molecules were lysophosphatidyl, especially those that have low number of carbons, C18-0, C16, and C18-1, and C18-2. This is particularly interesting. And next slide. Because we had previously found that those same metabolites were associated with greater decline of walking speed in older adults, still in the Baltimore longitudinal study of aging, and also was associated with lowered mitochondrial function that was measured by P31 spectroscopy at MRI. So this really suggested that the lysophosphatidyl had something to do with mobility, but also had something to do with mitochondrial function. The next slide. And the peculiarity of this lysophosphatidyl is the characteristic of the fatty acid associated with them. In fact, the 18-2, the 18-1, and the 18-0 are the most important constituent of a lipid that's called cardiolipin, that is specific of mitochondria, only exists in the subcrystal part of mitochondria, and is absolutely essential for the alienation of the complexes that assure, that the electrotransportation change is correct, and then there is a good function of mitochondria in good production of ATP. The next slide. So our future direction is to understand whether this parallel motor conjugated decline with age indicate a peculiar pathway to dementia. We're studying now what are the plasma protein metabolites that are associated with dual decline. We're starting with the Baltimore longitudinal study of aging, and we're trying to replicate this finding in other studies that are funded by NIA, such as, for example, Health ABC. And then we are also studying mitochondria to understand whether lower concentration of lysophosphatidyl in the plasma is associated with the decline of mitochondrial function. And in the future, we hope to study this directly in the brain by using 7T MRI. And then also we're doing a study where we look at the lipid composition of the brain of the individual with AD to see whether there is a decline of these same molecules. Thank you very much. It's truly an honor to be here. And I hope that you reach out to BLSA so that we can establish great collaboration between NIH and our institution.
Thank you. Thank you, Dr. Ferrucci. The pleasure is ours. As said in the beginning of the symposium, the goals of this symposium are threefold. First, to provide a forum to exchange current status and achievements of brain research. Second, to strengthen bilateral cooperation between Taiwan and the United States. And finally, to boost the innovative development of brain science. And so far, we have heard a lot of inspiring talks. Last, but definitely not least, we are honored to have Dr. Susan Ann Resnick, the Chief of Laboratory of Behavioral Neuroscience, NIH, to share longitudinal imaging in BLSA. Let's welcome Dr. Resnick.
Thank you very much for the invitation. And I know it's getting late in the evening for people in Baltimore and probably late in the morning for people in Taiwan. So I wanted to talk a little bit today about longitudinal neuroimaging projects, in particular, in our case, in the BLSA. Next slide. You heard from Dr. Ferrucci about the Baltimore Longitudinal Study of Aging. I wanted to note that since the mid-1980s, we've had a program of research on early markers of Alzheimer's disease, and more recently have also focused on predictors of cognitive resilience. So I was happy to hear that mentioned earlier. What I want to emphasize is that through the BLSA, participants come in for regular visits to the National Institute on Aging and receive a variety of behavioral and physical assessments. But since the mid-1980s, we've actually done prospective diagnoses of dementia. And we all know that trying to diagnose dementia retrospectively is really problematic because we're talking about people with memory issues. So one thing that's been really important is that we have a very long history of having these prospective diagnoses. Next slide, please. I came to the NIA longer than I want to admit ago to develop a brain imaging component as part of the early markers of Alzheimer's disease program. And I'd like to say that when we started this, people were very skeptical that you could actually use MRI in a longitudinal framework. But in 1994, we began imaging BLSA participants, initially close to about 160 individuals. And we measured brain volumes and lesions, white matter lesions in the early days. And then as the technology developed, we added other measures like diffusion tensor imaging, resting state fMRI, and some of the functional task-related MRI studies that you heard Dr. Go speak about, one of which he developed while he was in our laboratory. We've also had the opportunity to use positron emission tomography to look at, in the past, regional cerebral blood flow measures while people were resting and then during memory task challenges. But more recently, we've been using PET studies to look at Alzheimer's type neuropathology. Starting in 2005, amyloid imaging with carbon-11 PIB, and in 2016, fluorotaucipir to measure tau. And unfortunately, this program has been slowed a bit with the pandemic because we've only been seeing a very limited number of participants. Now, importantly, concurrently with all of these imaging measures, we do a full battery of cognitive testing. And historically on, close to 2,000 people, we have anywhere from 3,000 to 10,000 serial cognitive measures, depending on the particular sample and the time different tests were initiated. Next slide, please. It took us quite a while to be able to harmonize scans from different epochs, because one of the challenges in longitudinal imaging is that the scanners change and the technology changes. And it's always a balance between trying to maintain the older technology in order to maintain the longitudinal integrity. So you want to decide when you want to catch up with technology, but also try to compromise by maintaining the types of scanning parameters that you've been using. So recently, with our collaborators at the University of Pennsylvania, we've been applying multi-atlas techniques to measure regional brain volumes. And these techniques are really novel because they use a series of atlases and a series of algorithms to get the best match locally so that we can get accurate quantification of images longitudinally. And what I've shown you here are just a few of the regions: the total brain, the gray matter, the white matter, and the hippocampus. And each line represents an individual. And you can see that across the board, we see longitudinal declines in brain volumes, but individuals are measured very stably using these kinds of approaches. And this has been really important because it's allowed us to use data over up to 20 years of longitudinal trajectories looking at regional brain volumes. And next slide, please. Next slide. Thank you. What this has allowed us to do is to look backwards in time to identify the earliest brain changes that are associated with cognitive impairment. And with Nicole Armstrong, a former postdoc in our laboratory, we used the MRI data over this 20-year period, and we were able to identify 65 people out of 600 and close to 70 that developed cognitive impairment. But the novel aspect of this paradigm is we only use the MRI scans during timeframes when they were considered cognitively normal. So the important point here is that these people would be included in any cross-sectional study of aging. But yet we know that some point after the last MRI scan we use, they've developed impairment. And so we can look at the longitudinal trajectories of people with subsequent cognitive impairment and compare them to people who remain cognitively stable over time. And we were able to do this in a large group. The individuals were roughly around 70 years at baseline, about half men and women. They, had typical ApoE genotype frequencies. Of course, the subsequently impaired tended to be more likely to carry the E4 allele. Next slide, please. And what we do is we compare these people who go on to develop cognitive impairment to the people who remain cognitively normal. And what you can see is that when we ask the question, where do people who develop subsequent impairment show greater volumetric decline or greater ventricular enlargement, we have this plotted here. And the black dashed line is the 0.05 level of significance. The red dashed line is the 0.01 level of significance. And we can see that a number of the structures in the temporal lobe shows steeper volumetric loss in people who are going on to develop impairment, even though they're still cognitively normal. And we can see that there are differences in trajectories. Interestingly, most of the regions that showed significant differences in rates of volume loss were temporal lobe regions, not entirely unexpectedly. And we also see that, ventricular enlargement as sort of a gross measure was also greater in the subsequently impaired compared to the people who remain cognitively normal. Next slide, please. More recently, we've been able to move beyond these kinds of volumetric trajectories and to track Alzheimer's disease pathology over time. And I think that with the advent of the measures to look at beta amyloid accumulation and more recently tau accumulation, this has really revolutionized our ability to understand how people are changing with respect to Alzheimer's type pathology. What I have plotted on the left, higher scores here, these are distribution volume ratios, but they represent increasing amyloid. And you can see that there's quite a bit of scatter, even though the majority of these people are cognitively normal. But the picture becomes very clear when you add the longitudinal component. Once people are beyond this dashed line threshold, which is actually a very, very low level, they are on an upward trajectory and they don't go back down. So we can detect, using these kinds of longitudinal approaches, individuals who are just starting to accumulate amyloid pathology. Next slide, please. And this is important because amyloid is related to memory decline. With Murat Bilgel and Jacob Zients in our lab, in an initial paper, we looked at people who were either amyloid positive, had neurodegeneration as measured by hippocampal atrophy, and looked at combinations in relation to memory decline. And not surprisingly, the people who had both amyloidosis and neurodegeneration, hippocampal atrophy, were the ones who showed the steepest memory decline over time. More recently, we've looked at tau in the entorhinal cortex. And again, most of these people are cognitively normal, but despite that, we see that higher levels of entorhinal cortex tau measured by PET are associated with steeper trajectories of memory decline. Next slide, please. And I just want to point out that I've shown you some results from the Baltimore longitudinal study of aging, but I think one of the really important things that we've been able to do is participate in a number of consortia, and hopefully the relationship with Taiwan will grow in that direction. So two of the consortia that we work closely with are the iStaging Consortium, led by my longtime collaborator, Christos Davatsakos. And at this point, he has harmonized almost 20,000 MRIs across the age range. And it allows us to look at this issue of cognitive resilience and brain resilience, because we can look at typical aging with age and then look at people who either show advanced aging or resilient aging, show very little volume loss with age. And another collaboration to look at this period of preclinical Alzheimer's disease is the Preclinical Alzheimer's Consortium that Marilyn Albert leads, and BLSA is part of that consortium as well. And we're working with harmonized MRI, cognitive, clinical data, as well as PET data, to try to look at interactions between ApoE, between baseline age and other factors, lifestyle characteristics. And we need these kinds of larger samples to be able to tease apart some of these more complex interactions. Oops, sorry, I just hit my spacebar. But next slide is my next to last slide, please. And I just have a few comments. I think that longitudinal studies really present unique challenges. I've alluded to that. It's a balance between staying current with technology and maintaining the longitudinal integrity so that you could really look at change over time. Secondly, that by looking at these longitudinal trajectories, we can identify changes in the brain associated with later cognitive impairment, and it's clear that they're evident many years before symptom onset. I think that PET imaging and the new plasma biomarkers, which we haven't talked about today, are really tremendous in allowing us to test hypotheses in vivo now. So we can use these longitudinally, whereas before we could only look post-mortem at, as I like to say, which is necessarily cross-sectional. And these biomarkers, most of you have heard all of this in the news. The biomarkers have really been critical for patient selection into clinical trials and for therapeutic monitoring. It turned out that a lot of people who were being enrolled in anti-amyloid trials didn't have amyloid in the brain. So nowadays, you wouldn't do any of these studies without using some type of marker of the pathology you're trying to target. And my last slide is just to say that these are really complicated studies, and there are many people who've been involved in making this successful. I tried to keep it simple today because of the interest of time, but we have many sort of tangential studies that also are integrated into the BLSA program. And I want to thank Luigi for all of his support over the years and making this program a success. So thank you.
Thank you. Thank you, Dr. Resnick, for the remarkable presentation. After all the talks, the next 20 minutes will be joint discussion session. And for the participants, you could type your question in Teams meeting chat room. As a reminder, please state your name and respected title. Right before we start, I'd like to welcome Dr. Sun, our moderator, for joint discussion. With his expertise in molecular biology, I'm sure we'll be able to drive a lot of valuable exchanges today. Let's welcome Dr. Sun.
Good evening and good afternoon. I am Henry Sun. I'm from the Institute of Molecular Biology, Academia Sinica in Taiwan. I know it's late. We are about 30 minutes behind schedule. But this bilateral symposium, the purpose is to promote mutual understanding of our the research in Taiwan and the research in NIH, and hopefully to promote future collaboration. So I think the the discussion session is very important. So I hope our colleagues in the U.S. East Coast will endure for a few more minutes for our discussion. And now we're open, the floor is open for questioning. Any comments? I would like to first call upon Dr. Chi-Hong Lee, who is online, who used to be a PI in NIH, in NICHD, and he was recruited back to Taiwan and worked as a director in the Institute of Cellular and Organismal Biology in Academia Sinica. Chi-Hong?
Hello. Can everyone hear me?
Yes.
Okay, excellent. Okay, well, it's great to see old colleagues. Okay, I also saw the Alan Koreski was also online. Hello, Alan. As Henry mentioned, that I was a senior investigator at NIH and has moved back to Taiwan until about 3 years ago. And since then, I've been trying to promote a collaboration between NIH as well and Taiwan's neuroscientists. So we did host a joint symposium a few years back between Academia Sinica and NIH, where at the time was more focusing on synaptic physiology and molecular neuroscience. I would say that we do confirm our mutual interest and also complementary perspective as technical expertise. So I was thinking about, from the perspective of at the administration level, what can we really promote this? Okay, so I sort of draw from my experience at NIH for 17 years, what really helped me most and what helped to establish collaboration later on. And it dawned on me that I was really helped mostly with for example, the JSPS program, the visitor program that I'm sure that many of you know about it, that allow the Japanese scientists to join NIH with some financial support. And later on, there are also like a Korean equivalent, like the KVSTA, and so on and so forth. So I would imagine that would really be helpful to consider sort of a Taiwanese version of this fellowship program. You know, I would really urge the most to consider forming this and as a gateway for the Taiwanese neuroscientists to be able to actually come to work at NIH. The other aspect that I also find particularly useful was the visitor program. The NIH has this visitor program that I believe is still working, and they allow, for example, sabbatical research professors, national, international scholar to actually join NIH for a short period of time. Okay, so I would imagine that would be certainly useful for some form of bilateral effect. Obviously, Taiwan's we might be able to use the NIH visitor program for short visit, but I think would be advantageous to have some form of program to allow NIH research to come to Taiwan. So that's what I'm trying to say.
Okay, thank you, Chi-Hong. Do we have any other questions from the audience?
Can I just make a couple of comments in response to Dr. Lee? I think that we are all waiting that the pandemic relent and give us a little bit of breathe space so that we can start having direct scientific discussion face to face. Something that we all dramatically miss. At least I do. And of course, there are incredible opportunities for visiting scientists and collaboration. In particular, NIA, as Nina had shown, is now creating this new initiative that is called CARD, the Center for Alzheimer's Disease and Related Dementia, where we really invented new ways of collaborating with others, from the visiting scientists for short period to people that present collaborative project and can come to realize this project using the facility of CARD, to collaboration through multiple institutes. And also, we're trying to insert this is really a challenge given the structure of NIH, some collaboration with industry. So I think that there will be a lot of possibilities, but I will also invite you to consider that there are enormous amount of data that are already generated.
Yes.
And even without a direct contact with the visiting, now that it's really difficult, but just started by using reciprocally the data that we collected and share the idea that we have on the science could be incredibly useful. It can be incredibly productive and create a route that they can evolve in visiting scientists, reciprocally, U.S. scientists coming to visit you and some of you coming to visit us. Yes. I think that is a real possibility. And I want to finish by asking a question. I know that there has been, or there is already, a Taiwan longitudinal study of aging. I think that I collaborated with some scientists long time ago, and then I lost connection. Is somebody able to give us some idea what's happening there? Because there would be another opportunity for us to collaborate.
Thank you, Dr. Ferrucci. I think, are you referring to the Yangming Veteran Hospital longitudinal aging group? I think that's still ongoing, right? Can you? Okay.
Yes, that's exactly what I was talking about. I was in an advisory board years ago, and then, things evolve. Everybody goes in different direction, but it was a very, very interesting study with a lot of biomarkers collected, a lot of samples connected. It was many years ago, so at this point, the outcome should be there. And if this is still alive, that's certainly, especially in the research on biomarker, will be an incredible opportunity for collaboration.
Yes, thank you. I'm Chimpo Lin from Yangming University, and I guess who you mentioned maybe Liang Gongchen, who is the PI for our longitudinal data set. Actually, we have the data set for 10 years, and most of the elderly have MR scanner, clinical access, and also cognitive access. And nowadays, more focused on frailty, and recently we also nominate and all kinds of physical cognitive decline syndrome. I guess that's quite similar to what you talk about, the parallel physical function and cognitive decline with aging. I guess we have a lot of collaboration in the coming future, so I will take the message back to the team and see how we can link together for further studies. And also, by the way, my major is MRI, so we have a lot of imaging techniques. So I guess that's also quite highly interesting to me to use the data set, BLSA, and see how can we combine those data sets together and see how can we have further collaboration. Thank you.
It's a very good time to talk about new imaging in the BLSA because we're getting a new scanner, which means we're going to be reevaluating our protocol. Whenever we have to update scanners, that's the time that we try to figure out where we need to make changes.
Thank you, Susan. Any other comments? We have an online question from Sandy Chen, Taipei Medical University. In addition to aging and degeneration, we have collaborated with NIH for years in head trauma associated with neurodegeneration. We hope we can collaborate with NIA to broaden the scope. This program of head trauma is very traditional, has been ongoing for several years in the Taipei Medical University. and hopefully they can establish some collaboration with our friends in NIH. Any?
Mark Allett here. I can just comment that it might well be useful as well to have collaborative studies where investigators from Taiwan and the NIH can do a project together with sort of compatible expertise, or perhaps better said, different expertise. There are some times when projects in one place or the other require somebody who has additional expertise, and knowing what the opportunities are, what investigators are at each of the institutions can potentially help that. So activities such as what we did this evening can help inform what are the different investigators on the different sides of the Pacific Ocean here. And then there could well be money set aside in addition to visits for people from one to the other, but also longer-term collaborative projects.
You know, I think the other possibility that relates very much to what Mark just said is that particularly in the area of head trauma, we actually, in the extramural sphere, not on the NIH campus, but across many centers in the United States, fund a multi-center head trauma study that then each institute that is contributing to that deposits their standardized data into a common database. And so you really get the power of many institutions contributing data into a database that has agreed upon consensus-derived common data elements. Everybody measures things in exactly the same way. And I wonder if there would be something gained by harmonizing in some way the database and the study that you're talking about already ongoing in Taiwan with the existing database in the US, and thereby not only being able to compare and contrast, but being able to make N quite large as a function of the multi-centers involved.
Thanks very much for the comment. Any other comments, questions?
Yeah, I would like to echo this excellent idea. And because as I mentioned during my talk, I already mentioned that we do need a Taiwan comprehensive and longitudinal database. Although I have heard that Dr. Chen Lianggong has done a lot of work, and I actually heard his talk before, and probably he has collected more than 5,000 subjects or something like that. But it's a little bit difficult for me to sort of, because I'm a psychologist and focusing on a lot of cognitive functions. And because what I understand from Dr. Chen Gongliang's database, probably they use mostly about neurological testing or something like that. I don't know if I'm wrong, that Dr. Lin Qingpo can correct me. But I think it's needed to sort of extend the database across Taiwan. But of course, we need to respect whether or not Dr. Chen Gongliang was willing to share the database and extend it. And I really looking forward to collaborate with Professor Susan Resnick about the BLSA, because I think BLSA has a very long history, starting from 1958, earlier in my age. And I think this is really excellent. I mean, it really would help longitudinal and healthy scientific research in Taiwan. So I really think at this moment, I myself really encourage we should start thinking about this. And maybe this is a start point for the collaboration between NIH and the MOST. This is my comment. Sorry. Yeah, thank you.
Okay, is there any other comments?
I actually have a question, if I might. So I would like to know if across the universities there, they will have access to, the presentations today, the trainees, because the idea is that through these collaborations, we can exchange students, graduate students, undergraduates or postdocs, so that we can train them either in Taiwan or here, and then they can bring back to the countries, what they have learned. So I think that that's really important as well, because they are basically the driving force for these collaborations as well. So.
Okay, thank you. At the last, I want to emphasize that today we are presenting research topics from this Taiwan Brain Research Program. But in addition to that, we have many other neuroscientists whose projects are not in the special program. And all of these can be are also looking for collaboration with our U.S. friends. Okay. So now I would like to invite our Director General, Dr. Chen, back on stage to say a closing remark.
So first of all, I'd like to thank Henry for moderating the discussion section. I also like to thank all the speakers for inspiring talk and all the participants for valuable suggestions. So as I said in the opening remarks, today's symposium is just for our cooperation. So we can promote at least 16 for bilateral cooperation next year. Okay, so we also anticipate more personnel exchange to strengthen our partnership and also empower the brain technology innovation. So thank you again for all your participation, and wish you all the best. And we hope we can see you together in near future. Thank you. Thank you, everybody.
Thank you. Thank you, Dr. Chen, for giving us a wonderful closing. And once again, thank you, Dr. Sun, for hosting the joint discussion. And thank Dr. Lee, Dr. Zhong, Dr. Allen, and all the esteemed researchers to join the discussion. And this has brought today's symposium to the end. Again, we'd like to express our sincere appreciation to all of you for your presence in 2021 Bilateral Symposium on Brain Research. And if you have any questions or want to initiate a deeper discussion, please feel free to contact us through the email shown on the screen later. Hope you enjoyed the symposium today. Thank you and goodbye.